And I think we're recording now. It is the Wednesday evening session for our week 8, I believe. So it's the review week for the exam and practical exam 2. Before I make a start, I just wanted to check, is there anything in particular that I can help with? Do you have any particular questions, or you're good with just a review as we've been doing normally? If you have something in particular, you just write me a message or just unmute yourself and let me know. Otherwise, what I'll do, I'll make a start with the review session because, like I said, I don't want to keep you super long today. I want to get through the material so you get also to your evening and spending the evening. For anyone who's watching the recording, just for the record, I had a morning session that I recorded, but it looks like that recording got corrupted as part of the saving process, so I don't have that available, unfortunately. And for the Monday morning and Monday evening, no one joined me. I was here by myself, so I didn't record myself chatting away. So hopefully this recording will work and it will be available, uploaded on the learning module as well. If something were to happen, I have recordings from previous semesters, but I would prefer to do it with you guys with the recording that's for this class. So let's have a look of what we've got lined up for this session. And what I really want to spend most of our time is to go through the study guide for the exam 2. Of course, I have these other things that I'm mentioning here as well that we'll quickly talk. But first and foremost, what I wanted to share with you, can you believe next week is spring break? I feel like I'm more excited than anyone else. So next week, we'll actually be taking time off. So what I've got listed there, the weeks, they're wrong. They all should be 1 week behind. So don't look at those. Next week, we're not going to have class. We're taking a spring break. And then when we come back, we move on to the respiratory system. But by the time when you are finished with this week, you have completed half of the course. You also have completed almost half of the points towards your final grade. So I think that that's always exciting to realize that we're that far. This week, there's no new assignments. Only thing that I'm asking you to do is to complete exam 2 and practical 2. If you have any missing assignments or you need any extensions, I would really encourage you to use this week for that. You just message me and let me know the details of assignment that you need either opened or extended, because I would ask you to please try to get all the missing work as far as you can by the end of the spring break. So when we come back from the spring break, I will be closing the previous assignments unless I have made other arrangements with you. And after that, those assignments are closed. So I'll have to start entering zeros to the assignments that if someone didn't complete those, because otherwise your grade calculation that you see automatically updating, it's not going to be correctly reflected. But even if I mark 0 and you want to later on attempt that, you just have to let me know and I'll then remove that 0. But try to get missing assignments as far as you can done by the end of the spring break. But as always, if you need extra time, all you have to do is message me. So that was just a quick reminder of that. Like I said, this week's material, the exam 2 and lab practical 2, they're due at the end of the week, 1 minute before the midnight on the night between Sunday and Monday. So if you can, please try to get those done. They don't take a long time unless you have an accommodation plan. Each one takes 60 minutes. So that's all the time that you need to schedule for those. So just to give you a heads up, that make sure that you get those in. I have prepared a study guide for the exam 2, and the exam 2 is the same format as it was with the previous exam. It's an open book exam. You can take it from home and there is no proctoring. So you do not need to set up the any sort of proctoring software or anything. You can do it without any of that. What you will notice that all of the questions for the exam are from this study guide. So it is really good idea to either join me like you did, thank you, or if you didn't have a chance to join me in person, at least watch this recording, because that's the only place where I give answers for the missing gaps on that study guide. And on that note, if you don't have the study guide available, I encourage you to go to our learning module or this week's module, and you'll end up noticing that a study guide is there. So let me just pull up the screen. I should be sharing my internet browser with you. And when you go here to the modules, I'm just going to jump to the student view. Seems like our canvas is running a little slow today, but we're getting there. So when you go to the modules and you scroll down to week 8, you'll see that I have there a study guide for exam 2 covering heart, blood vessels, and lymphatic and immune system. When you click in there, that's where you are able to download this study guide. And like I said, there's a lot of blanks in there. And these blanks are for the reason that you either need to join me or then you just need to be really clever and work them out. But I give out answers to those missing blank parts on these revision sessions. So that's why students either join me in person or then students watch this recording. And every single question, like I said, in the exam is from this study guide. So that's all I wanted to say about that. Let me just jump back to our PowerPoint. So what we are going to do, we're actually going to move on in a few seconds to go through this study guide. So if you didn't have a chance to download it from the learning module, like I just showed you, this would be a good time to do it. If you didn't have a chance to print it and you want to print it, you can do that. Or if you just want to have it on the screen and take your notes as we go through that, you can do that as well. But because I know that we have a lot to cover, before I move on to talk about the study guide, I will just address everything else that, I didn't mean to do that. I will address everything else that I have in this session for you. So we will go through all of the material. So by the time we're finished with the study guide, you can just log out. I want to remind you that for the lab practical, I also have these guides that show the common terms in terms of the anatomy that you want to know. So these terms are structures that you want to be able to identify those from those pictures that you've been covering as you have been completing your practice anatomy sessions. So it might be any of these that I ask in the practical exam. And I really, really, really encourage you to go again to week 8 learning module and find this practice version of the practical, because by completing this practice version, you'll get to see all the possible questions that might come up in your actual practical exam. But because there's only so many questions that we can fit into this practical, it pulls up these questions from a larger pool of questions. So I encourage you to try it a couple of times, several times if you wish, so you get a chance to see every possible question that will come up. So you might want to do it 2, 3, even 4 times. It's up to you how many times you feel that you need to go through it. And remember, the practical as well is going to be open book, so you can take screenshots, you can take notes, some students even use their mobile phone to take pictures as they go through those practical. There is no proctoring. There is a time limit, so you go through those, prepare to those, and you will ace it. I also have uploaded handouts of the lecture slides in case you want to use those while you're revising for the exam. They're not the exact same slides that I use in my lecture videos. That's because of the copyright issues, but these are the slides from your textbook. So they will help you, and it will cover all the same material. Finally, I want to remind you that we do offer tutoring, even though we're not meeting in person. You still can have tutoring. You can do our online tutoring that's available through the college. That was called the Cranium Café. You just log in. The web address is posted everywhere possible, and you don't need to have an appointment. You just show up there, and based on what's the availability of the tutors, you pick the tutor that you want to have a session with. If you have a favorite tutor, you just wait until they're free. If they're free, they will see you immediately. If they're seeing another student, then you just get placed into the lobby where you wait until they're free. The other option is that, of course, you can also contact me. I'm more than happy to do tutoring sessions, and in fact, over the past few weeks, I've done quite a few of those in this class for students. So you let me know that you would like to have an appointment with me, and I will schedule it. Usually I can schedule it, if not for the same day, then at least for the following day. So I'm fairly open for any requests. And as always, if you have questions, if you're attending to this session like you are, I'm happy to facilitate those at any stage. So you can just unmute yourself and give me a shout out, or you can type it to the chat, and I will be so happy to answer anything. If you're following or joining me on this session through a recording, you can always message me. I check my messages on the Canvas every few hours, but I do also check the email, but the Canvas is really the quickest way to get hold of me. And that's really all that I have planned for this session. So what we are going to do without further ado, we're going to move on to the study guide, and I want to discuss the 3 chapters that we will have covered in this exam, or that this exam will cover. So let me just jump out of this presentation. And if you were able to download that study guide or print it or have it on your screen, great. If not, that's not something that you have to do. It's just an extra resource that I offer. I just use it to guide me so I remember to talk about everything that I possibly want to talk about with you guys. So let me share my screen for that. And at this point, you should be seeing my PDF file viewer. So the Adobe, it should be visible for you. And we will go through the material by using that. Again, like I said, at any stage, if you have questions, you just shout out. So we're going to start with the chapter on the heart. I think the hardest chapter that students are most stressed about is going to be the chapter on lymphatic and immune system. But I promise that I don't ask that many questions from that system. And the questions that I ask are very general. So we're going to make a start. And as the very first thing, look at the heart. And we're going to cover both heart anatomy and heart physiology. So whenever we talk about anything to do with the heart or structures that are related to hearts, we talk about cardiac field. We talk about cardiology, or if you talk about cardiac doctor, that's a heart doctor, or if you have someone who does science, where he or she looks at heart or related structures, that person would be doing cardiac science. So that's the term that you might see being used sometimes. And as we start looking at the heart, I just want to connect what we're talking about to this bigger picture of the cardiovascular system. So there's going to be two circuits that I want to discuss with you. So let's go through those. So the first circuit that we're going to look at is pulmonary circuit. And you might remember that pulmonary circuit was this circuit that transported the blood from the heart to the lungs. So that's why pulmonary circuit was an important one for us, that it's all about getting blood from the heart to the lungs and bringing it back from the lungs. So let's have a look of that circuit together. So what I'm going to do here, I'm going to draw a really naive picture of a heart, and we'll learn all about heart later on. But that's our heart. And blood leaves the heart to the lungs. So let's add the lungs here. These are supposed to be a drawing of lungs with its two sides and trachea coming out. The blood that leaves the heart and goes to the lungs is gonna be deoxygenated. This means that there's very little oxygen in it. So it's poor in its oxygen content. Instead, this blood carries a lot of carbon dioxide. Well, once this blood now has been at the lungs, it comes back from the lungs as reoxygenated blood. Now this blood is rich in oxygen, so its color changes actually to brighter red color. And this blood is now nice and rich in oxygen, so it's something that our body really, really wants to use. So this, what we just looked at, is our pulmonary loop. And we're going to continue our discussion and have a loop of the second loop that we have for the blood. And this second loop is going to be called, let's see, this looks better. It's going to be called our systemic loop. This blood that leaves from the heart to the systemic loop, it goes to the rest of the body. And now, since it's just this blood has came back from the heart, it is rich in oxygen. So it's going to be oxygenated blood. Again, you remember a lot of oxygen here. And then this blood is supplied to the tissues, which use the blood from there, or use the oxygen from there. And after the blood returns back to the heart from the body, now all the oxygen has been used and it is deoxygenated blood. So there's only little oxygen left in this blood. So this was our systemic circuit or systemic loop. So we saw that on there, and it returns back as deoxygenated. If you have any questions about this, give me a shout out. Otherwise, I'll carry on. If you are watching the recording and you have questions, you can always wind back. Just let me know if I can help with anything. So that was setting our kind of thoughts of what the heart is, where is it in this bigger cardiovascular setting. And now what we're going to do next, we're going to actually talk about where the heart is located. So this is a great chance for us to practice proper medical terminology so that we actually are able to use the correct terms to describe the heart and where it is placed in our body. So what I'm going to do here, I'm drawing a heart. And really, if you think of the heart, it is a little bit of a cone-shaped structure. So I'm trying to draw it accordingly. And we'll talk about this cone shape a little more, but you'll notice that the top part is a little larger and at the bottom it has this little bump there. So where is the heart located? Well, first of all, when we talk about directions, remember we always talk about directions in relation to the patient's right and left. So this is going to be the right and this is going to be left. Well, the heart is located within your chest cavity. And rather than saying the chest cavity, of course, the term that we use to describe that is going to be our thoracic cavity. So heart is located within the thoracic cavity. Another term that you'll see being used is that the heart is located within the mediastinum. Mediastinum was this place, this space between the lungs. So that contains only the heart and the major vessels that come in and out of the heart. What else can we use to describe where the heart is located? Well, let's use this color. Heart is located between the lungs. So we can add our lungs here. So that's a good positioning term for us to kind of appreciate the location of the heart. Another term that you see me using here is that heart is located above. So that would be the term superior to diaphragm. And diaphragm is this thin, large muscle sheet that separates the thoracic cavity from the abdominal cavity. And that is the main respiratory muscle when we move on to talk about the respiratory system. The other kind of a clue, which is especially good clue when we look at the surface anatomy, is that the heart is located between the ribs 2 and 5. So let's add our ribs here. So that's our first rib, and that's our second rib, third, fourth, and fifth. So heart is located between second and fifth rib. So if you ever have to position, for example, any ECG LEDs on someone's chest and you're not quite sure where the heart is located, you can simply feel where the first rib is and then count down from there. The heart is between the second and fifth rib. And now we get to the part that I mentioned to you that we can describe heart as being a little bit of a cone-shaped structure. So I'm going to draw on top of what I just showed. There's the heart there, this cone, what it would look like. So you'll notice that it's got this sharp end. And this, if we use this, we can actually describe heart's location really well. So let's draw a line to the middle of the body. And we'll notice that the heart is located slightly on the left-hand side of the body. And remember, it's going to be left of the patient. It is slightly off from the midline, which we just drew as the dotted line. And if we look at a structure called apex, the term apex refers to this tip of this cone. Well, apex actually is pointing downwards. So that's the term inferiorly. And it's pointing to the left, and it's also pointing to the front. So the term for that would be anteriorly. So these would be all very good ways of describing the location of the heart. And I could see myself asking that in the exam, that pick the statements that are correct describing the location of the heart. Now, if we look at the heart is actually not, the heart muscle is not just one layer. It's made of multiple layers, and heart is also enclosed inside a sac. So let's have a look of all this. So what I'm drawing here is the heart wall, as if we would be looking on a microscopic level of that. So this all is the heart's muscle wall. So let's add that to this diagram of ours. And if you want to have kind of a landmarks, what we've got on this side would be the inside, and this side would be outside. So we can divide actually this muscle wall of the heart into 3 layers. The innermost layer is gonna be our endocardium. So endocardium is this innermost layer that we see. And the outermost muscle layer is going to be called epicardium. And in between those, we've got the thickest part, which is myocardium. So those are the 3 layers, and we're looking them in an order of moving from inside of the heart to the outside. So that was the heart wall and the 3 layers of that I wanted to discuss with you. Oh, I didn't mean to do that. This looks better. So our endocardium, the innermost, middle thick layer, myocardium, and outermost layer, epicardium. Well, heart is also located inside this cavity, inside this connective tissue sac. And we're going to draw it here. It's got inner layer, and it's also got outer layer. So this innermost layer is our visceral layer, and outer layer is our parietal layer. And in between the visceral, our inner connective tissue layer that's surrounding the heart, and parietal layer, which is the outer layer that surrounds the heart. In between those, we've got this space. And this space is known as pericardial cavity. So it's filled with pericardial fluid, and that one is going to have a very specific purpose, which we will discuss as the next thing. And the purpose of having fluid in between these 2 layers of pericardium is that by having fluid in there, it works as a lubrication, so allowing these layers to slide past each other much nicer. So why would you put heart inside this pericardial sac? I think that would be a very good question. Why on earth do we put heart inside this sac? Well, this has 2 purposes. First of all, you remember that the heart was located between the lungs. You remember? Well, because it's located between the lungs, it could be that heart gets really squashed in there and it would be hard for the heart to move. So by putting it inside this sac, we kind of make space for it, that the lungs are no longer contracting it. So it has space for the heart to move and do its job. So that's our lungs. The other reason why we put a heart inside a sac is the same reason why we see all of these separated cavities in the body, is that allows us to separate important organs from the rest of the body, so that if there's an inflammation somewhere, it's not going to be able to spread in to these important structures of ours. Let's say that there's an inflammation here in our thoracic cavity, and inflammation spreads easily. But now, because the heart is inside this sac, it's not able to go and get the heart inflamed, which would be really, really bad news for us if it got inflamed. So that's the reason why we got the heart inside this sac. And inflammation in medical language was always itis. And if it's the inflammation of the pericardium, that's of course going to be pericarditis. And you can actually hear this pericarditis with just a stethoscope. You will hear that, oh, there's this crackling sound, so something's not wrong. And that would be an issue for the heart's pumping ability. Well, let's move onwards and have a look of the muscle tissue that the heart is made of. So you remember we had 3 types of muscle tissue. We had our skeletal muscle, which was these big muscles that we see that cause movement in our body. Then we had smooth muscle, and we'll talk about smooth muscle later on, but smooth muscle is mainly found in the intestinal tract, kind of forming sphincters that separates different parts, and also in around our blood vessels. And then we have, as the third one, this cardiac muscle tissue. So let's look at a single cardiac muscle cell. Which of these statements apply to that? Well, first of all, cardiac muscle cells are branched. So they have multiple branches out of that cell. The other thing that we see is that they have striations that run through this cell. These stripes, like a zebra-like stripes, they resulted from the fact that the muscle fibers slide past each other, and wherever they're crossing over, that shows up darker in a microscope, and wherever they're not crossing over, that shows up brighter in a microscope. In addition, in a heart muscle cell, we've got one nucleus, so we call it uninucleated cell, and that nucleus is normally located right in the middle. And of course, the only place where we find cardiac muscle cells is going to be in the heart. Now comes the last term that's important. You'll notice that at the end of these branches of a heart muscle cell, we've got this specialized structure that's called intercalated disc. These intercalated discs are something that's going to be really important when we think of the function of the heart and how it works and what requirements we have for its work. So that's why we end up finding them. So I'm drawing a couple of other heart muscle cells, and you'll notice that one cell connects to another through this intercalated disc. Well, intercalated discs allow materials and also messages to pass from one cell to another easily. So what we want to have with the heart muscle cells is that they all contract at the same time, because this is how we get this big uniform contraction of the heart. So we want to pass messages from one cell to another, and it is these intercalated discs that allow us to do that. Well, next thing that we're going to do, we're going to talk about the flow of the blood through the heart. And we're going to also talk about different chambers that we have in the heart. And we're also going to talk about different valves that we have in the heart. So let's have a look of all of this. And like I told you earlier on when we first looked at the heart, that the simplest way of drawing the heart is by drawing a box that you divide into 4 parts. That's your heart. Each one of these smaller boxes is a specific chamber. So let's name those chambers. First, we have on the right side of the patient, the more superior chamber. This is gonna be our right atrium. Then underneath the right atrium, so inferior to that, on the right side, we've got right ventricle. And on the left-hand side, we've got the same thing. Left atrium, more superiorly, and left ventricle, more inferiorly. So atrias are always the more superior ones, and ventricles are the more inferior ones. And then you just add whether it is right or left. That seems quite easy and quite good for us so far, right? Well, let's add the flow of the blood to this diagram. And blood comes to the heart either through inferior vena cava or superior vena cava. Inferior and superior vena cavas drain either the superior drains the upper limbs, head, and neck, whereas the inferior vena cava drains the blood from all other parts of the body. And this blood enters to our right atrium, and from there it crosses to our right ventricle. But right atrium and right ventricle are actually separated from each other. And they are separated by a valve that's located here. And that valve is either going to be called right AV valve or tricuspid valve. The tricuspid stands for the fact that it's made of 3 leaflets. So valve's function was to control that the blood can flow only one way and one way only. So blood will always flow from right atrium to right ventricle, never the other way around. You also notice that this blood is now deoxygenated, so it has very little oxygen in it. It's coming from the body. Well, we're going to send this blood to lungs. And to get to the lungs, it leaves from the right ventricle and passes through one more valve. And this valve that it passes through is gonna be our pulmonary valve. So pulmonary valve makes sure that blood can move only from right ventricle to the pulmonary trunk. And pulmonary trunk then divides into 2 branches and further and goes to the lungs. Well, now that blood is coming back from the lungs, it's going to be rich in oxygen. So it's now reoxygenated or oxygenated blood. So that's something that our body can use. And it's coming back from the lungs to the left atrium. And there's actually 4 different branches that bring the blood to the left atrium. But for our purposes, we just know that it comes to the left atrium. And to move from left atrium to the left ventricle, this blood has to pass through another set of valves, which are located here. And they are known either as left AV valve or bicuspid valve. You remember the bi stands for that there's now 2 cusps here. The third name that you sometimes see being used is the mitral valve. So it can be any of those names that people use when they discuss this valve. Well, after that, the blood leaves the left ventricle and travels to the aorta. But to leave the left ventricle and move on to the aorta, it has to go through one more valve. And this valve is going to be called aortic valve. And now we saw that the blood travels to aorta, which is the first big artery leaving the heart. From there, it travels to the body, where it gives off the oxygen and returns as deoxygenated blood back to the right atrium. So this was the body loop, was our systemic circulation. We've talked about that earlier on. That's when it goes to the body. And the loop that brought the blood to the lungs was our pulmonary circulation. So that makes sense as this kind of schematic diagram. Let's now add a little bit more reality to our drawing and look at it on a diagram that's more realistic. What you will notice though is that we can still see our four chambers there. We can see our right atrium here. We agree on that. We can see our left atrium here. And we can see our right ventricle and the left ventricle. So all the same stuff that we saw earlier. So now let's go through the flow of the blood, and we're going to do it a little bit quicker because we've now looked at this already. So the blood is coming to the right atrium through inferior vena cava and superior vena cava. This blood is going to be oxygen poor, so deoxygenated. It leaves from the right atrium and travels to the right ventricle. And as it travels through from the right atrium to right ventricle, it has to go through this pair of valves here. And valves had a function of making sure that material could travel only one way and one way only. And this valve is known as right AV valve or the tricuspid valve. And you remember, again, the word tri referred to the three cusps that it's made of. Now, blood is ready to leave our heart, and it leaves from the right ventricle and goes to the lungs. And as it does this travel, it has to go through one more set of valves. And these valves here are gonna be our pulmonary valves. And after pulmonary valves, there's a pulmonary trunk, and then we have our lungs. Now the blood that returns from the lungs, it comes back to the heart, and this blood is going to be rich in oxygen. So it's oxygenated blood. And it enters to the left atrium. From the left atrium, it travels to the left ventricle, and this valve is going to be known as our left AV valve, or as bicuspid valve, based on the number of cusps, or mitral valve. So we can add that to our diagram here. And finally, the blood leaves our left ventricle and travels to the rest of the body through aorta. And to do this crossing from the left ventricle to the aorta, there's one more set of valves. It's here. and that's called our aortic valve. And again, as the blood travels now to the body, it gives up its oxygen and it returns back to the heart as deoxygenated blood. Couple of other structures that I want to add to this diagram. First of all, earlier on, we talked about the kind of a cone shape of the heart. Well, the very tip of the heart is called apex. So we saw it here as this tip, the apex. And apex is a term that can be used for other structures than just the heart. You'll notice when we talk about the lungs have apex, which is the sharp part, but it's located somewhere totally different. Another term that I like to ask from the students is this term here, interventricular septum. Septum is a wall of some kind, and when it's interventricular, it's located between the ventricles. Okay, we now have covered largely everything that we need to know of that. Just a couple of terms that I want to make sure that everyone knows. Here I'm drawing the valve. And valves had a function of making sure that material can pass through those, but the material can pass to only one direction and one direction only. If it tries to return back, it's not able to go there. So that's how the valves serve purpose of making sure that the flow of blood is unidirectional. And it is the pressure changes that either open or close the valves. So as the chamber contracts, the pressure grows, and eventually this pressure is enough for the valve to open. If we have a valve that has some problems with this, we can have a valve that is not correctly closing, so we're going to have backflow. Blood flows back where it shouldn't. In this case, we just end up moving this blood back and forth. It's not very good. The other option is that we have a narrowed valve, so there is insufficient opening. So the valve doesn't open enough for all of the blood to flow easily through it, and as a result, the heart has to now contract even more forcefully, and that's a problem as well. Couple of other terms that I want to show when we talk about structures of the heart. If I have here, again, a drawing of the heart, and we remember that we could divide it into 4 chambers. More superiorly, we have atrium, and more inferiorly, we had ventricles. Well, atria actually have, when you look at the surface of the heart, it has this kind of a flabby little part on its top surface. This is known as auricle. This flabby little part gives extra space, extra volume when the atria fills up. So we can fit in more blood there, but when the blood is, there's no blood in the atria, auricles collapse. Few other terms, we end up seeing inside the ventricles on the wall this adaptation that they're kind of wiggly on the inside of the heart. These wiggly parts are known as trapeculæ carneae, and their function is to make sure that blood doesn't end up pooling anywhere there. So all the blood gets squashed out. Because if we have blood somewhere that's not constantly moved, it starts to clot, and we don't want blood clots forming. And finally, the term septum, we already talked about that, separating 2 parts. Okay, you can have sometimes heart valves failing. If you have a heart valve that fails, we deal with that by either installing a mechanical valve or a valve from another animal. For example, pigs are very common, that we take heart valves from pigs and then we give it to humans. or we can go and find a dead person and give a cavitary valve for that patient. Couple of other terms that you'll see. Fossa ovalis refers to this small opening in the embryonic heart where we don't have lungs that would function because there's no air to breathe inside the mother's womb. So the blood flows directly from right atria to left atria. Coronary sinus is what returns the blood from the coronary circulation, so supply of the heart valve, and it returns it here to the right atria. And ligamentum arteriosum is another embryonic remain bypassing the lungs, coming from the pulmonary arch or pulmonary trunk, and bringing the blood directly from there back to the heart. So it doesn't do that loop to the lungs. A little bit of surface anatomy, what you need to know. The term sulcus should be something that you're familiar from when we discussed about the brain in BIO201. Sulcus was this kind of a depression on the surface of a structure. So we had those sausage look of the surface of the brain. The depressions, deep ones, were either fissures or sulcus. Well, we find those depressions also on the surface of the heart. So let's draw our heart here. And there's a couple of depressions that we end up seeing. So first of all, there's our 4 chambers. And let's start with our atrioventricular sulcus or atrioventricular groove. Atrioventricular groove separates, of course, our atria from our ventricles. So it is this groove here. In addition, we have interventricular sulcus. It separates the ventricles. And we have one on the anterior side and one on the posterior side. These are important anatomical landmarks because they are what we use to explain where the coronary arteries are located. So let's go through the coronary arteries. So first of all, we have our left coronary artery, which arises right after the aortic valve and divides into 2 branches. The first branch is known as anterior interventricular artery, or sometimes also referred as left anterior descending. So it's our anterior interventricular artery. The name tells where it's going to run, so let's add it here. It runs on the anterior side between the ventricles, so on this groove here. That's that. The second branch that the left divides is left circumflex artery. And left circumflex artery runs in this atrioventricular groove and then wraps around to the back of the heart. In addition to that, we have another branch of the coronary arteries, and this branch is going to be our right coronary artery. So that's the third branch, and that runs between the right-hand side, between the atria and ventricles, so atrioventricular groove, and again, it wraps to the back of the heart. So those are the coronary arteries, and those are the grooves where they run. Okay, couple of words about the physiology of the heart. Heart contractions are involuntary, of course, so you don't have to think that now my heart is going to contract, and now it's going to contract. And
Can you go back, please?
Sorry?
Can you go back to the last page?
Of course. This one or even further?
Further.
Okay,
Yeah.
this one?
Yeah. No, the other one. The one
So
after
now-
this.
Would you like me to explain it again, or you're good with that?
No, the one after this. I just need to see it. I didn't get
Okay, sorry,
everything.
I went on too fast. I appreciate you.
Yeah, you did.
So this was supposed to be on green color. I think it's just
No, the other one after this.
this one.
Yeah, this one. Yeah.
Perfect. Yeah, I know there's a lot of text. So basically what I'm telling is that the anterior interventricular artery runs in that groove that was labeled as B in the previous table, and then the left circumflex runs in the groove that was labeled as A, and the right coronary artery runs in the groove that's labeled as A, but it runs on the other side. You're good? You got those?
No, I'm
Take your time. There's no rush. I'm not rushing you.
Trying to like fill in the gap.
I know there's so much going on this.
I ran out of ink, so I couldn't print it out. So.
Oh, I'm so sorry. Take your
you're fine.
So you just let me know. I so appreciate that you shout out. Sometimes I go through it so fast because this is actually what I used to do for a living. So cardiac anatomy is my field. So that's why I go through it fast, because I think that it makes sense, but I realize that not everyone has done it forever. It's their field.
Yeah, that's true. Now I got it now.
Perfect. So let's look at how the heart works a little bit. And what we noticed here, that the heart contractions are involuntary. That means that your heart will contract without you having to think about it. It just happens on its own. So there is this kind of internal, just natural beating rhythm for the heart. And we'll talk about that in a second. But before we do that, I want to show this diagram about the membrane potential on the heart, and a couple of words that I just wanted you to match in there. Let's start with our depolarization. Depolarization, you remember from when we talked about the nerve cells and action potentials on them, is when you are approaching the zero membrane potential and going even above it. The repolarization referred going back to being polar, so being back far away from zero. And finally, there's this resting phase, which is called pacemaker potential. And you remember that in a traditional nerve cell action potential, these were looking a little different than what we're seeing with the heart. And we'll talk about the difference in a second. But those would be the three terms that I would want you to match to that diagram. And again, let me know if I go too fast. So what we're going to do next, we're going to look at the heart surface and how these electrical currents pass through the heart. So the heart pacemaker that generates the heartbeat is called SA node. And it's located here on the superior part of the right atrium. That's what generates the heart's beating rhythm. From there, an electrical current will travel all across the atria, both on the left and right hand side. And as this electrical current travels through, it causes the atria to contract. After that, there is this delay station called AV node, which causes this electrical impulse to be held still for a second. And this is so that the atria have time to fully contract. Once the contraction of atria is completed, this electrical current continues down in our interventricular septum. You remember that. And it goes down what is known as bundle branches, then Purkinje branches, and finally wraps around the ventricles. And once it ends up wrapping around the ventricles, that's when the ventricles contract. So you'll notice there's more steps for it to reach around the ventricles so that we have time for the blood to flow from the atria to ventricles. So heart is always beating in this rhythm. Atria contract, ventricles are relaxed, ventricles contract, and atria are relaxed. So it goes that other one is always contracted, other one is relaxed, so that the blood can flow out from whatever is contracted. Let me know if I rush too much. So let's have a look of now a couple of clinical considerations. If this heart rhythm, which I showed you, was that other one is contracted, other one is relaxed, it's not regular. It's kind of wherever it happens to be. There's no this nice rhythm. We talk about the patient having arrhythmia. It's an irregular heart rhythm. And sometimes the type of arrhythmia that we see is fibrillation. In fibrillation, there's no pattern whatsoever. It's just going totally crazy. And this would be problematic because now blood is not pumped correctly. So your heart has gone into fibrillation. What you can do to treat a patient whose heart has gone into fibrillation, you take those 2 electric pads and you give them a shock. You can also do it as a regular lay person by using a machine called AED, automated external defibrillator. So that gives your heart an electric shock, and the hope is that the electric shock stops this weird contraction and re-establishes this normal contraction pattern. So we just give them a shock to bring them back to that. Some individuals actually have fibrillation happening all the time. So then we don't take those machines and give a shock. What we do instead, we implant a small machine on their chest, which is called a pacemaker. And that gives their heart the shock whenever it needs the shock to recouple the atria and ventricles so they contract together. Let's talk a little bit about the heart rate regulation. Heart rate is regulated from the brain in a part called medulla oblongata. And I think you know the medulla from your previous study on BIO201. And from there, the signal travels down the spinal cord to this SA node that we saw earlier on. And the SA node was the pacemaker of the heart. If you have heart rate slowing down, that's usually down to the vagus nerve. You remember that from the cranial nerves that you covered also in BIO201. Finally, I want to talk about the action potential of the cardiac muscle cell. So let's look through that quickly. You remember this looks very similar to what we saw with the nerve cells action potential. However, this is just much longer. It takes a longer time for the cardiac muscle cells action potential to travel. And this is because we want to give time for the blood to flow from one chamber to another. So let's notice these parts. Depolarization was again going towards 0 and even above it. Plateau was this stable state of electrical current. And repolarization was becoming polar again, away from 0. But what you'll notice is that this takes longer than in a nerve cell so that the blood has time to flow. Then we talked about the ECG a little bit. That was the electrocardiogram, sometimes called also EKG. And what we did with electrocardiogram was that we put a bunch of LEDs on patients' chests. And that allows us to kind of count the amount of electrical currents, how they summate in the heart. And usually the typical EKG trace looks like this. So we have that drawn there. And let's look at different parts of that. So the first pump that you see is going to be called T wave. That tells us that now SA node has depolarized and atria has depolarized. After that, we had this shooting down, shooting up, shooting down, and returning back to normal. This is known as our QRS complex. During this time, the ventricles depolarize and atria repolarize. And finally, we have our T wave during which the ventricles repolarize. So you should know the P, Q, R, S, and T from this EKG trace. And another thing that we look, in addition to looking that do these look normal, so we want to see that are they all where they should be, and do we see all of these events, and are they right shape? We can also look distances between these different events, so the intervals between them to establish if that's a healthy EKG or ECG. Couple of more terms that I want to talk. We already earlier on talked about the fact that some of the cardiac chambers were always contracted, so they're in systole, whereas others are relaxed, so they're in diastole. Systole refers to the contraction, so now the chamber is coming as small as it can, so it squeezes the blood out, whereas diastole refers that is a dilated chamber. It's relaxed, so it can receive the blood. So other parts, atria or ventricle, is always contracted, and the opposite part is diastole, in a diastole. So it's relaxed. So those would be good terms to know for the exam. And a single cardiac cycle is one complete heart cycle covering all of these events that we have looked. Next, we can hear heart sounds when we listen to heart with a stethoscope. And these heart sounds tell us about how well the heart valves are working. So the first heart sound that we end up hearing is often described as this lub sound, and it tells us that our AV valves have now closed. So when these valves slam closed, they actually make this sound that we can hear. And the second sound that we'll hear is the sound produced by the pulmonary and aortic valves closing. So pulmonary and aortic valves are known together as semilunar valves. So semilunar just means pulmonary and aortic valve. Finally, about the cardiac output, heart puts out about 5¼ liters per minute. And that's a sum of multiplying the stroke volume, so how much heart squeezes in one contraction, by the number of contractions that you have in a minute, which is our heart rate. But what's noticeable is that this can go up very, very high when you're exercising, when you're working hard, up to 4 times, 5 times of the normal amount of blood pumped in a minute. So when we're exercising, our heart works much harder. So I wanted to say a couple of words about the clinical relevance. That's the congestive heart failure. That's something that you can study independently, same as the embryology. They're not going to be in the exam. But I want us to go through quickly different blood vessel types. And we'll notice that we follow these always in the order of how the blood flows away from the heart and back to the heart. So after the heart, we've got our arteries. These are the vessels carrying blood away from the heart. After that, we've got smaller versions of arteries. They are known as arterioles. After that, we've got capillaries. And then we have venules, which are smaller versions of veins, and eventually veins. What I want you to notice from here is that it is within the capillaries where the exchange of materials happens. So oxygen and carbon dioxide, as well as nutrients and waste products, are exchanged between the capillary and outside of the capillary, so the cells outside. Okay, that brings us to have a look of the general structure of the blood vessel, and we're going to move from the innermost structure of the blood vessel. So we're going to move layer by layer, looking at what we can see that makes up the blood vessel. In general, what are the typical layers? And we'll work our way from the inside towards the outside. The center of the blood vessel is known as lumen. So let's do just that. The innermost layer is known as tunica intima. So it would be this layer. And how I think of it is that it's intimate because it's in intimate contact with the blood inside the blood vessel. That's made of simple squamous epithelium. So one layer of these flattened disc-like cells. And they are simple because one layer and squamous that they're flattened. They are this shape so that it's easy for the materials to pass through this layer. Next layer is going to be our tunica media. Let's add that to our diagram here. This layer here. And tunica media is mainly made of smooth muscle. There's some elastin there. And this smooth muscle allows us to change the diameter of the blood vessel based on what needs we have for that blood vessel. So if we are doing vasoconstriction, that means that the diameter constricts, it goes down, it decreases. Vasodilation instead refers that the diameter of the vessel increases. The final layer that we're going to have a look of is going to be the outermost layer of the blood vessel, this layer that we see here. And this layer is known as tunica externa, sometimes also called tunica adventitia, depending on where in the world you are. I think tunica adventitia is more common in Europe and in UK. So externa, it's the most outermost. That layer is made of mainly connective tissue, and it really just anchors everything to its place, reinforces the walls, supports the walls. Sometimes we have blood vessels that have such a thick wall, such as here, that it is this outermost layer, tunica externa, that has hard time receiving the blood supply because every tissue in the body requires blood supply. So we don't have an issue with getting blood to the tunica intima or tunica media. That's short enough distance for the blood to travel from inside the blood vessel. But this outermost layer, which we saw as tunica adventitia or tunica externa, that's going to have hard time to get the blood. So we have a special adaptation to supply blood in these thick, big vessels. And that's going to be called vasa vasorum. Vasa vasorum is simply a blood vessels that come from the blood vessel itself and supply this outermost layer here. Okay, that brings us, let's have a look at the arteries in general. Arteries are exposed to high pressure, so that's why they're going to be very thick and elastic, so they can handle this huge pressure fluctuation. And their function is really to smoothen out this pulsatile flow that comes out of the heart. And there's going to be a few different kinds of arteries. Let's go through those. And the first ones that we are going to look at are going to be our elastic arteries. The name really tells about how their structure. They are made of mainly elastin, and they are sometimes also called conducting arteries. And this conducting tells that they really just transport blood from one part to another. They're a conduction pathway. There's no vasoconstriction. All they do is that they expand and recoil, but they never shrink smaller. So their function is really to even out that pressure that comes out of the heart with the blood being pumped in a pulsatile flow. Then we get to the muscular arteries. And muscular arteries are instead described as distributing arteries. So they choose where the blood goes rather than the conducting arteries, which just simply transport it. And actually, most of the arteries are muscular arteries. So what we end up finding is that there's going to be a lot of smooth muscle in these walls of the muscular arteries, very little elastic tissue. And they can do now this vasoconstriction that we didn't see earlier on. So they can constrict if we don't want blood going to a certain part. The vessels that lead to that part get constricted. And the final kind of arteries that I'm going to mention is going to be our arterioles. They're simply smaller versions of arteries, just a single layer of thickness right before we get to the capillaries. And what you remember that we have more capillaries in the body than we actually have blood. So we can't have all of the capillaries open all at once because that would be too much for the blood. There's not enough blood for that. So what we see here is an arteriole coming to a capillary bed, and then capillaries on the other side as well, going to a venule that leads to a vein. But not all capillaries are open at all time. We choose which capillaries are useful for us and which ones are not. Where do we want to direct the blood to? And this is done by these pre-capillary sphincters that I've shown here. So they open and close their bands of smooth muscle and choose where the blood should go and where it should not go. So that's how we choose only part of the capillary networks to be open. Okay, next we're going to talk about the capillaries. Capillaries are the smallest and thinnest blood vessels in the body. So there needs to be a very, very, very low blood pressure there. Their thin wall would otherwise explode if the pressure was very high. It's tunica intima, the only layer that you find there. And the function why you find only tunica intima was that we had this exchange of materials that we saw earlier on here. That stuff is able to cross back and forth at the capillaries. So it's the main site of exchange of the materials at the capillaries. There's three types of capillaries that I want to mention to you. The first one is continuous capillaries. And what you'll notice about the continuous capillaries is that the cell wall of these, all of the cells are connected. So it's a continuous layer. There's some examples where we find them at the skin, muscles, lungs, central nervous system. The next.
Can you go back, please? Sorry. Can you go back?
Further back?
Yeah.
To here?
No, it's fine. It's right there. Yeah, it's
Take
fine.
your time. So these were the
No, go back.
sphincters. They opened and closed, choosing which of these capillaries are open.
Okay, you can go. You can continue now.
Good. So the continuous capillaries were this layer of wall. All of these were continuously connected to each other. The second type of capillaries that we find is going to be fenestrated capillaries. In fenestrated capillaries, we have fenestrations, which is just a French word for window. So small windows here and there. So you see them here and there. They are little openings on the cells that make up the walls of the capillary. You find these, for example, at the kidneys, intestines, sites where hormones are secreted. The final one is going to be sinusoidal capillaries. Sinusoidals, you think of the word sinus, means empty space. They're missing large portions of the walls of the capillaries. This causes the blood flow to be very slow, and materials are able to cross in and out of the capillary very, very easily. So we find this, for example, at liver, bone marrow, spleen, adrenal medulla, and so on. So those were the three types of capillaries. capillaries. And we already talked about the capillary bed and the precapillary sphincters. Capillary bed is just a network of capillaries, and these precapillary sphincters were those muscle bands that we saw here that opened and closed. So I've listed it twice, maybe because it's important topic. Okay, let's have a look at our veins. So veins are the vessels that bring the blood back to the heart, and blood pressure in the veins is going to be very low. And actually, majority of your blood vessels are going to be veins. And we talked about how it is hard to get blood back from your toes to your heart. And this was hard because we're going against the gravity. So it's hard to bring in that blood. And this was facilitated in a process called venous return by having valves that we see here, just like heart valves that allow the blood to flow only to one direction and one direction only. So the blood is able to go only to this direction, and then if it tries to go back, these valves prevent the flow. Veins get constantly squashed by the skeletal muscles. So we have veins are constantly squeezed, and every time that a vein gets squashed, the blood moves then up to the direction where the valves facilitate them to go. So you'll notice that was the concept of the venous return. And for example, soldiers who stand with their knees locked, that's why they end up passing out, because they're not getting the blood returning back to the heart. One final term that I'm going to introduce here is going to be the term of anastomosis. Anastomosis just refers to these connections between arterioles and venules. So there's a bypass not going all the way through this capillary network that we saw. Okie dokie. I want to mention a couple of words about the blood pressure. So blood pressure tells us about the force that we have from inside the blood vessel being pushed against the wall of the blood vessel. So there's our blood vessel again. And the blood exerts a force against that wall. And that's what we call our blood pressure. And blood pressure is measured as millimeters of mercury. So when you ask from a person what's a normal blood pressure, typically the answer that you hear is something like 120/80. It changes a little bit from year to year and depending on what resource you're referring to, but something around that range. So why do you have two numbers for this force? Well, the first number is the number of the force during contraction, so the systolic number, and the second number is diastolic number, so the force during relaxation. So those are the two numbers that we get. And there's many different things that affect your blood pressure. For example, the amount of the blood in the system. If you lose a lot of blood, your blood pressure drops. Also, how much your heart is putting out blood, so cardiac output, the resistance to the blood flow, and also the elasticity of the vessels. Some of us, when we get our blood pressure measured in a doctor's office, it's higher than what it normally would be. And this is because we just get stressed out when we see a doctor. This is known as a white coat syndrome. So your blood pressure just appears unusually high because you're a little nervous of getting it taken. Okay, final thing that I want to discuss is our pulse. Pulse tells us the number of beats that the heart beats per minute. In other words, that tells us our heart rate. So when your ventricles push the blood out, we get the blood pressure shoots up. And when ventricles relax, the blood pressure drops. So you can tell from this variation the number of heartbeats. And you can measure it in a number of places. Carotid pulse from the neck, which is the most common. Ulnar pulse from the pinky side at the wrist. Radial pulse from the thumb side on the wrist, brachial pulse measured from the cubital fossa, so on the other side of the elbow, popliteal pulse measured from the leg. You can measure posterior tibial or dorsal pedis pulse from the feet, femoral pulse from the groin, and superior temporal pulse and external maxillary pulse from the temple of your head or your skull. We discussed about the short-term regulation of the blood pressure using this diagram of the homeostatic feedback mechanism loop. We saw that the blood pressure was off. That was noticed by baroreceptor reflex or chemoreceptors. They're located at the carotid sinus, aortic arch, and large vessels. That information gets passed to the medulla of the brain, and if it is anything else than 120/90, then effector kicks in. And we can deal with that either through medication, treating the cause, or having vasodilation or constriction happening. So that's the short-term regulation of blood pressure. Long-term regulation is done with the kidneys. And there's two different, so kidneys function really is to regulate the blood pressure. And there's two ways of doing it, direct mechanism and indirect. The indirect is also known as renin-angiotensin-aldosterone mechanism. That's all you need to know. So basically, if you want to summarize what you need to know about the blood pressure, we want to have high enough blood pressure that the tissues are supplied blood with, but not so high that it's going to break the blood vessels. And then you notice that we had a situation where we had too high blood pressure, that was hypertension. And then we had hypotension where you had too low blood pressure. And this could be due to temporary low blood pressure. For example, when you stand up, you get a little dizzy. You can be chronically low blood pressure, so often related to poor nutrition, or it can be acute as a sign that you're going into a shock. And we are going to go through the signs and symptoms of the shock. And the mnemonic for that was PC fats. So police constables that are fat. So if someone's gonna go to the shock, their skin is pale in color, it feels cold and clammy, their breathing is fast and shallow, their pulse is gonna be fast and weak, they might appear agitated, irritated, and they are often thirsty. And that means that they're gonna go into the shock. So that would be a good one to know for the exam, the signs and symptoms of the shock. I think that would be a nice question. And finally, we're going to look at the special circulations. So most of the blood normally goes to the brain, about 20% to the skeletal muscles, but that can go up if you're exercising. Your skin receives blood to regulate your body temperature, and it's also a great place to store blood. Lungs must have a low blood pressure so that they don't get punctured, and we talked about the heart earlier on. Then I've offered you a bunch of different kinds of diagrams that really show where the blood vessels are, what are the major ones are. So use those. Remember, the exam is open book. You want to know those. And finally, I want to mention that let's look at first of all, the speed of the blood. Blood travels fastest after the heart at the aorta and arteries. The speed drops by the time we get to the capillaries and then picks up a little bit after that. This is because we want the blood to travel really slow once we are at the capillaries, because you remember capillaries were the site where the materials got exchanged. And if we look at the total diameter or total volume, total space, cross-sectional area of the blood vessels, we notice that most of that is at the capillaries again. We want to have a lot of cross-sectional area there because that is the site of exchanging the materials. Finally, we saw that there were two forces that worked to push blood out of the vessel and blood back to the vessel, hydrostatic and osmotic pressure. And if those were not in balance, we end up with edema, more liquid being built up outside the blood vessel. So if you want to summarize what you need to know based on those diagrams that I offered you, a bunch of different ones, the big take-home message from them is that arteries typically run deep, whereas veins are superficial. And that completes that. And then we have the lymphatic and immune system. And really what I wanted to show here was that the lymphatic and immune system, they are linked with the circulatory system as well. So all of these systems work together. So we saw if we had liquid that traveled within the blood vessel, that was blood. And the part of the liquid that leaves the blood vessel and goes out, that is going to be the plasma from the blood. But once it has left the blood vessel, we no longer call it plasma, although it's the exact same thing. Now we call it interstitial fluid. And once the interstitial fluid gets picked up by the lymphatic system, we no longer call it interstitial fluid, even though it is the exact same thing, we call it now lymph. So if we compare and contrast lymphatic and cardiovascular system, the big thing to remember there was that the lymphatic system was open from one end. So it's not a closed loop like cardiovascular system. And in terms of the functions of the lymphatic system, the biggest one was to fight against diseases that we do with white blood cells. The other one is to transport fluids so that we don't end up with edema, too much fluid somewhere. And the last one is something that sometimes surprises students. So if you eat fatty food, your capillaries at the cardiovascular system are not able to pick up those fat molecules in your digestive system, but it's going to be actually the lymphatic system that picks up the dietary fats, the large molecules. So lymphatic system helps us to transport them, and it transports the fat molecules and then dumps them to the cardiovascular system right before the heart. So let's have a look of the lymphatic vessels. We have, again, just like we had in the cardiovascular system, we have lymphatic capillaries. They are much more permeable than the blood vessels were, or capillaries in the blood vessels. They are made of simple squamous endothelia. We already saw these flattened discs of one layer. And the purpose of this is to make sure that the passage of the materials across them is easy. Well, then once the lymph has been picked up by the capillaries, we now get the various different kinds of vessels that we have. They grow in size as we move onwards. The difference between the blood vessels is that they're much more thinner and they have these valves, just like the veins of the blood vessels. And I mentioned a couple of examples there. You can check them out from the lecture video. So bigger than our capillaries in terms of the lymphatic vessels are going to be known as trunks. They are named based on the location: lumbar at the lower back, intestinal trunk at the intestines, intercostal trunk between the ribs, bronchomediastinal trunk between the mediastinum and bronchi, subclavian trunk below the clavicle, and jugular trunk at the neck thoracic region. Bigger vessels than trunks are going to be our ducts. And there's two. We've got our right lymphatic duct, and we've got our thoracic duct. So if you want to relate these to each other, just like we did with the blood vessels, you would see that the capillaries were the smallest vessels. And once multiple capillaries join together, we end up with a trunk. And once multiple trunks join together, we end up with our ducts. So it's going bigger and bigger and bigger. And then eventually they dump all of their contents to the blood vessels by the heart. Lymph, in terms of what is lymph made of? Well, lymph is made of all the same stuff as your plasma, with the exception that plasma, of course, doesn't have white blood cells. So we have water, we have proteins, and we have white blood cells. One thing that you should never see in the lymph is red blood cells. So if you look at the lymph, it's kind of a whitish, milky color, opaque, transparent. But we should never see red in that. And lymph was produced in the bone marrow and lymph nodes. Well, we saw if the lymph ends up building up somewhere, we form an edema. If it's formed because of the lymph, it's going to be lymphedema. And causes why lymph might be ending up building is somewhere that the blood vessels are squashed closed by tumors, tissue growth, or they have been removed, some of those lymphatic vessels. And we're going to be looking at the T cells and B cells when we talk about the immune system. So those are the lymphatic cells that we'll talk about. Finally, I want to mention about the lymph nodes. Lymph nodes are these kind of bean-shaped structures. There's hundreds and hundreds of them in the body, and they're often present as clusters around a single lymphatic vessel. So you can think of them as a pearl necklace. And a lymph node, their function is to filter the lymph, and that's also where the immune system cells live. So what immune system cells do, they fight against any pathogens that get into your body that shouldn't be there. And when they're doing that, we have inflammation happening. And while you have inflammation, your lymph nodes swell. So that's why the doctor tries your neck and tests whether they're swollen and also if they're sensitive to touch. Well, lymph nodes are named based on their location. Cervical lymph nodes at the neck, axillary lymph nodes at the armpits, supratrochlear lymph nodes above the trochlear notch here, inguinal lymph nodes at the groin region, abdominal lymph nodes at the abdomen, and thoracic lymph nodes at the thorax. The easiest accessible ones are the cervical and axillary ones. Lymph nodes have both cortex and medulla, and they have various sinuses inside them. And sinuses are where these immune system cells live. Let's have a look of the lymphatic vessel as the next one. And lymphatic vessel, what you want to know of that we've got vessels coming in on this larger concave surface. These vessels are known as afferent because they come into the lymph node. And the vessels that leave the lymph node, they are leaving from the smaller concave surface. And they are known as efferent because they're exiting the lymph node. If you have swollen lymph nodes, that's typically an indication that there is some kind of an infection going on. And that's what the doctor was checking for. We went through a few different lymphatic organs. The spleen was probably the biggest lymphatic organ, about a fist size, located at the left side of the abdominal cavity. So if we draw a quick diagram, here is esophagus, your stomach, small intestines, and this is liver here. This is the right side, this is the left side. So our spleen would be located right here. Function of the spleen is that it filters the blood and it removes any old or dead blood cells, platelets, and in embryo, that's where the red blood cells are being made. And what you noticed if you studied those diagrams of the blood vessels, you noticed that we had a splenic artery and splenic vein. So it's rich blood supply to the spleen. The next lymphatic organ that I want to talk about is the tonsils. So we have many tonsils depending on the side where we see them. Palatine tonsils are the posterior oral cavity, so at the back of your mouth cavity. Lingual tonsils at the base of the tongue. Pharyngeal tonsils at the posterior nasopharynx. Those are the ones that we see easily. And tubal tonsils where the auditory tube connects to the ear from the oral cavity. If you have an inflammation of tonsils, of course, we talk about itis. So in this case, tonsillitis. And if you have repeated inflammation of the tonsils, you might need to have those removed surgically. That would be tonsillectomy. The next structure that I want to talk about is going to be your appendix. That's the first part of the large intestines right after cecum, and you can get that inflamed and that gets removed. So you can live perfectly fine without appendix. So you ask me, why do you have it then? It's probably that it's just an evolutionary remain from when we ate more plant-based food. So that's where the bacteria to digest plant-based food were housed. But now it's not used for us. So it keeps shrinking and shrinking, and I think eventually will disappear. And finally, thymus. You remember we talked about thymus earlier on. That's where the T cells went to mature, and it was larger when you were younger and then shrank because it got replaced by connective tissue and adipose tissue. In terms of the immune system, what you need to know, we're keeping it very, very simple. So rather than being a structural system, it's actually all about function. It is housed within the lymphatic system. So we really don't have anything to talk about its structure. So let's have a look of the immune system. What we want to focus there is going to be the white blood cells. So white blood cells are the immune system cells, and their function is to protect us against pathogens. And if you ask me what a pathogen is a disease-causing invader to our body. And so it's something that should not be in our body. There's things that make us sick, and there's many different kinds. They can be viruses, bacteria, parasites, fungus, all sorts of things. How they make us sick is that they get into the body and then they start multiplying in numbers, and that's what makes us sick. And there's two branches to the immune system, and we're going to go through both of them. So the first branch of the immune system is our innate immunity. Innate immunity is all about having general responses, and it really doesn't care against what kind of a pathogen it deals with. Innate immunity works through different lines of defenses, and the first line of defense is our physical barriers. So say, for example, your skin. Skin stops those pathogens from entering into your body. So it's a physical barrier. We don't have skin inside our body, but the lining of the oral cavity, nasal cavity, intestines, and from the other end as well, that's mucous membrane. So that as well prevents these pathogens from entering to your body. If we don't have a physical barrier that fails, then we have chemical line of defense. So sweat kills a lot of pathogens, so that's why they die on the skin. Also, if you eat something you shouldn't eat, the stomach acid will kill those. Your eyes are really easy route for things to enter to your body, so that's why tears are great chemical to kill pathogens, and also urine kills pathogens. We also talked about different cell responses, but as you see here, I just list them, so I'm not going to ask about those in the exam. There's 4 or 5 different signs and symptoms of the inflammation, and these I do want you to know. So if you have a site that's inflamed, it is going to be warm because a lot of blood is flowing to this site, and the blood is coming from the core, so it brings temperature with it. It's going to be red as well because, again, a lot of blood is coming to that site and we can see those red blood cells. This site is also going to be swollen because as blood flows in there, there's more material and the site needs to expand to fit in all that material. And it's going to be painful because there's so much stuff that the tissues are being pulled apart. So those are signs and symptoms of inflammation. Sometimes we also add loss of function. as a sign and symptom. Fever was abnormally high body temperature. And then the second branch of the immune system that I'm just going to keep very simple, that's going to be our adaptive branch. In UK and Europe, we call it acquired immunity, which I think makes more sense. So adaptive immunity is achieved when you have met a pathogen once and then you meet it for the second time. So adaptive immunity requires you to have met that pathogen before. So you develop a memory against it. So think of, there's the pathogen that we meet once. We can't deal with that with our adaptive immunity. We need to recruit our innate immunity to kill that. But now if we meet it for the second time here, we already have met it once, and now our adaptive immunity can work to deal with it. So we need to develop an immunological memory against it. So that's why it is very specific. It targets only certain antigens, the antigens that you have met before, certain pathogens. The benefit of that is that it acts throughout your entire body, not just at the site which gets inflamed. It acts throughout your entire body. What you need to know about the next listing is that there's the B cells and T cells that do the responses in adaptive immune system, and that can be naturally or artificially. Naturally means that you met the pathogen. The artificial one would be, for example, our vaccines. With COVID vaccines, a good example of that. And then I listed different immunodeficiencies, autoimmune diseases, and hypersensitivities. Know each of those that which are examples of which. And that's all you need to know for this exam. I know that it was a longer session than I had planned. Do you have any questions about anything that we covered? Did I go through it too fast?
No, you're fine. I just wanted to ask the practicals.
Yes.
Is it all about like the practice anatomy?
Yeah, it's all about those questions that you've seen in seen in the practice anatomy. And what I really recommend.
So the lab practicals, like the practice one.
Yes.
Is it also the same thing with the ones we did before?
Yes, it is the exact same. So you type in the answer, I go through them again. I really, really, really recommend the best way to prepare for the practical exam is to go through this practice version of the lab practical, because if you go
that's what I'm saying. It's the lab practical, like the practice version. Is it also what we did during our lab anatomy practicals too, like for each week?
Yes.
For the 3 weeks, I mean.
Yeah, that's correct. So that's it. And
because I went through some questions and I noticed they were like the same one we did.
Yes.
Like the homework. So I'm just trying to make sure.
No, you're absolutely correct. What I recommend, remember, this is an open book practical, so you can take pictures, you can take notes, you can take screenshots. Some of my students even use their phone to take pictures. Anything that helps you, it's all up for grabs. There's no proctoring.
Okay.
Perfect. Is there anything else I can help with?
Nothing.
Well, I really appreciate you joining me today. I hope that if you have any questions, don't hesitate to message me. But this is all that I really have to share. You did good. I think that study guide is going to help you for the exam.
Yeah, and the pictures and the study guides, are they like in the exam?
I don't think that I have pictures too much on the exam. I think I have one where you are asked to identify the interventricular septum maybe, or apex, but that's about it. I think I have a diagram where I have the box diagram of the heart, and then I asked that which chambers would they be.
Oh, okay.
That sort of
Just wanted to know that too.
nothing super hard. And remember that you have the diagram next to you. I don't think I asked anything about action potentials or things like that. Maybe I asked that,
Okay.
why is it slower or is it slower in the heart? But that's it. That nervous system.
Oh, okay.
Okay, that's all I can think of. But if you have any questions, ask away. Otherwise, I wish you a very good rest of the evening and thank you again for joining.
You're welcome. Have a good night too.
Have a good night. Thanks. Bye. So that completes our recording of this week's material as well. As I said, contact me if you have any questions. But on this note, I hope you have a good week and enjoy your spring break. Bye.